One of the big challenges that we have faced as a field quite broadly is the difficulty in making accurate in vitro models that captured the disease process. And there are a number of reasons that that's been challenging, but potentially the biggest one is the fact that the human brain is really inaccessible to us during life. So up until the availability of iPS cells, we didn't really have a method by which we could have unlimited human neurons to grow in the lab. So in that regard, the availability of iPS has been really transformative. And so what I'm saying iPS without defining it, induced pluripotent stem cells. Obviously, the big advantage of this is we can make patient-specific models because we generate the stem cells from a sample of skin, which is easily accessible to us and non-invasive. And so in our lab, what we do is collaborate with our clinicians who are just in the building across the square. When they see a person with either a genotype or a phenotype of interest, they ask them for a skin sample, which we can then use to make their stem cells. The next step is making the relevant cell type from those stem cells. And there are two real ways that we do that. So we can make cortical neurons as a 2D culture. So these are cells which kind of growing in a single monolayer on plastic. We do this by a process called dual SMAD inhibition. So we're using small molecules that kind of essentially inhibit signaling pathways, and the consequence of that is the cells undergo something that very closely mimics cortical differentiation during human development. So these cells are really useful to work with. They've kind of given us a lot of insight into molecular mechanisms. They are more amenable than the 3D cultures that I will talk about in a second to things like drug screening, because obviously there's a monolayer. If we put a small compound into the media, there's a uniform exposure of those cells to that compound. However, there are some limitations, and one of the limitations of the 2D approach is, of course, because we're growing the cells in a monolayer, we're disrupting the architecture and the structure of the cells. So they aren't organized, for example, into layers like we would see the neurons organized in the brain. And so to try and address that, we complement our 2D models by using these 3D systems called cerebral organoids. It's a very similar principle in that we are mimicking the very early stages of development of the brain. However, this time we're doing it in 3D. So these are non-adherent, they're floating cultures, probably about the size of a lentil. And as you will have seen from some of the images I used, the advantage of this talk is you get different cell types occupying kind of different spatial regions of the organoid. They also, of course, come with limitations. I mean, in both systems, they're missing cell types such as microglia, they're missing a vasculature. The cerebral organoids tend to be a bit more variable compared to the 2D cultures, which tend to be quite homogeneous. And so that's the reason that we use both approaches to complement one another.